Our next presentation will be on melon severe mosaic virus. We included this one in the Thrips-transmitted section, but I will give the disclaimer that there has not been any confirmed Thrips transmission in cucurbits for this virus, but I will talk a little more about that as we go on. So, melon severe mosaic virus, MSMV, it's also been referred to as MESMV in various published research, Orthotospivirus melitessalati. This virus was first identified as a new virus in cucurbits in Mexico in 2007. It was found to be associated with disease that had an estimated yield loss of 20 to 30% in honeydew melon. And since that research, there had been no reported occurrences outside of Mexico, where it is now considered to be widespread, until 2022 and 2023. So, in general, there is very little published research regarding this virus. In terms of the United States, it's currently limited in the US. The first report in the United States was in Arkansas, where it was reported to be present in the fall of 2023. But research that we did in Mississippi indicated that the first known occurrence of the virus actually was in the fall of 2022 in a commercial pumpkin field in North Mississippi. So, as far as current literature reports go, it is reported in 2 counties in each of Arkansas and in Mississippi. I do know that there have been additional detections in other counties in Arkansas, and we have seen it in a couple of other counties in Mississippi. And not just a one-off occurrence, but the virus has been detected in either consecutive years or after 2 years of the first detection in some of those counties. And so we are seeing it not just as a one-off occurrence. So, in terms of symptoms, just as some of the other speakers mentioned about mosaic symptoms, melon severe mosaic virus does cause mosaics or mottles on the leaves as well as the fruit. Blistering and deformation of leaves is also possible, as well as cracking or splitting of fruit. And what we have seen in some of the instances in Mississippi are these frilly leaf edges that you may see here, as well as an alligator skin type pattern. So these are some examples of those frilly leaf edges, and in the image on the right, kind of what we are describing as that alligator type pattern. And we have seen various symptoms. The images in this plant, we can say that high throughput sequencing was used to determine the potential viruses present in this plant, and melon severe mosaic was the only land virus that was present in this plant. So we're pretty confident that these are the symptoms caused by infection with that virus. Another plant in that same field had somewhat subdued symptoms in comparison, and this plant also had a lower virus titer. So symptom development may be, or expression rather, may be dependent on that virus titer. Additionally, what we've seen is variations of symptoms, which you see here are some additional symptoms of plants that were also determined to be infected with melon severe mosaic virus, either through a serological lateral flow test or through RT-PCR or RT-qPCR. So it looks like there may be a variation in symptoms that may depend on host as well as the variety and the virus titer. So we're still trying to determine what the whole catalog of symptom expression may be. But one thing that you probably have noticed is that these symptoms are closely related or nearly identical to many of the other discussed widely prevalent mosaic-inducing viruses in the United States. The aphid-transmitted viruses, cucumber mosaic virus, papaya ring spot virus, watermelon mosaic virus, zucchini yellow mosaic virus, as well as squash mosaic virus. So symptoms alone cannot be used to identify virus infections, and as some of our other speakers mentioned, many of these also occur in mixed infections. There are some preliminary data to indicate that Melon severe mosaic virus may have occurred in a mixed infection with another mosaic virus, but that has yet to be confirmed. And so at this point, that is still preliminary. That being said, in terms of potential epidemiological factors, the host range has been investigated using either field surveys and diagnostic testing, as well as some mechanical inoculation tests. In terms of those field surveys and diagnostic testing, several of the economically important cucurbit crops have been identified to be infected with melon severe mosaic virus. And in terms of the mechanical inoculation testing, that was done initially with the detection of this virus in Mexico. They've seen several tobacco relatives, as well as the red tassel flower that has multiple different common names. Each of those various species were shown to have systemic infections following the mechanical inoculation. So it does appear that the virus can go to non-cucurbit hosts. Again, this is not a comprehensive list because there has been limited research done in terms of host range investigations. In terms of virus transmission, as I previously mentioned, a vector has not yet been confirmed, but because melon severe mosaic virus is an orthotaspovirus, we do suspect that there is a Thrips vector, one or more species. This is based on the fact that known Thrips vectors of taspoviruses, or I guess I should say that all of the taspoviruses that have been well characterized do have one or more Thrips Thrips species vectors as that are relevant to their spread and transmission. And in each of those viruses, the Thrips vectors do acquire the viruses as larvae and they transmit the viruses as adults, and that is done in a persistent propagative manner where the virus circulates and replicates in the vector. Additionally, in terms of mechanical transmission, this was able to be done experimentally, but that may only be successful for a few passages. One thing I do want to note in terms of the vector transmission and the suspected Thrips vectors, as Sudeep mentioned, there are numerous Thrips species that occur in cucurbit fields throughout the United States. And in terms of the potential vectoring of the virus, this would be the first persistent propagative virus confirmed, Thrips-transmitted persistent propagative virus confirmed for cucurbits in the United States. So, melon severe mosaic virus. Again, this is an orthotaspovirus. It is a single-stranded RNA virus that is comprised of 3 RNAs, RNA-S, RNA-M, and RNA-L. And if you are looking to try and confirm this virus in a new area, detection of all 3 RNAs is recommended. The full-length virus genome of one of the isolates from Mexico is publicly available in various sequence databases. And so that information is there in order to help with research. But talking about diagnostics, in terms of the early research, microscopy, ELISA, as well as Western blots were used to confirm and investigate the virus. But currently, lateral flow test strips that may be commercially available, as well as molecular methods such as RT-PCR, qRT-PCR, or high-throughput sequencing are the ones that are commonly used. There are various protocols that are now published with the RT-PCR and qRT-PCR melon severe mosaic specific primers that are available. And another option for confirmation, if it is paired with DNA or nucleic acid sequencing, would be to use RT-PCR primers that focus on, or that target rather, the orthotaspovirus more broadly, and then use sequencing to determine the species. So in terms of management, management options are limited, and this is largely due to the lack of knowledge that we have on this virus. Um, there are no known resources, sorry, there are no known sources of resistance to melon severe mosaic virus. And so much as what you've heard many of our other speakers talk about today, one of the most critical things that you can do right now is to initially scout for those virus symptoms. Um, as I mentioned, the geographic range is currently limited. So if you do see suspect symptoms, I would submit those to the appropriate diagnostic lab for testing, contact those labs to see what it is that they can do, and try and determine where the incidence of this virus occurs. Beyond that, removing and destroying infected plants, particularly if the virus is not known to occur in an area, may help to prevent establishment and potential non-cucurbit host reservoirs. And then beyond that, attempting to suppress thrips populations. As I mentioned, the one of the larval stages is the stage that acquires the virus, and then the adults transmit the virus. So trying to break that reproductive cycle of the thrips and trying to suppress thrips populations may also help to reduce virus spread. But again, there's a lot that we need to conduct research on to try and fine-tune management methods and determine which are most effective. So with that, I want to thank you for listening to me talk about melon severe mosaic virus. And I will go on to say that that was the last presentation for the formal presentations. And so that wraps up our presentations for webinar number 2.